some nice flower power. Okay, this is a simplified nucleoid. You know, the nucleoid is a big watered up hole like this. It's much bigger than this. But I have to draw it small so you can see what I'm doing. I can't work in that whole big thing. And don't forget, you got all your little hydrogen bonds holding the loops together. Oh gosh, I'm getting sloppy with my dotted lines. Whatever. Okay, here's my cell's DNA. Remember, it's the genome. If there's a plasmid, plasmid counts too as part of the genome. But we're not going to focus on the plasmid, we're just going to focus on the nucleoid, okay? Now, let's get back to her question, whoever that was.
What about, you know, if you have...
Starts and stops.
Yeah,
Let me get a couple colors here. Okay, starts and stops. So, how many genes are in a bacterial cell, first of all?
many.
Not that many. Compared to humans, not that many.
Well,
A plasmid can have, say, 5, 10 genes. Bacteria can have some, maybe as few as 100 genes, but probably up to like 3,000. Does that sound right, Colton? I'm never good with numbers. If I was good with numbers, I'd probably be rich today. But I can't remember numbers. I don't have a mind for numbers. Faces I can remember, but not numbers. And how many genes do we have? Well, they tell you in base pairs. We've got like billions. But at any rate, I'm only going to draw a few genes. My point is I can't put 1,000 genes on here, okay? But every gene has a promoter and a terminator. And the promoter and terminator is actually a sequence of letters in the DNA, which is a chemical molecule. So what I do for a promoter and the terminator, instead of putting the letters, I just put a block like that. So that would be the promoter, say for gene 1. And gene 1 has a functional part that says put leucine first, alanine second, valine third, threonine fourth, whatever. This is the functional part of the gene. So it might be the functional gene for, say, lactase, an enzyme, something like that. And then it's going to have another sequence that signals the end. That's the terminator for gene 1. Okay? I'm not going to write it for every single one. Just know that then there's going to be a promoter for gene 2 and a terminator for gene 2. Then we'll put a third one, promoter for gene 3, terminator for gene 3, and then don't forget you have your functional part and your functional part. And we'll do one more, gene 4, okay. Why did I bother to do all of this? Because in transcription, the cell is going to make an RNA copy of a gene, one gene at a time. Not really. You probably need multiple proteins at overlapping times. The cell's DNA is going to be busy. We're going to photocopy this recipe and make that protein. And we're going to photocopy this recipe and we're going to make that protein. And we're going to photocopy this. But in order to explain it, it's easier to look at it one gene at a time. Yes, Amanda?
is it true that in bacteria, unlike in prokaryotes, the eukaryotes, excuse me, it can have translation, transcription at the same time?
Probably. More complicated cells, yeah. And
Because we
different things are happening at different times because the cell has to function. And depending on what signals are there, it might make it start transcription for this one while it's still doing translation for that one. And sometimes certain proteins have to interact with each other. So you have to make this one and then that one. Did that help? These are, what's the matter?
Yes.
It did?
The thing is, what I was learning biology, okay, in my case, is that if we take, for instance, a human, how they make proteins, is that the translation and can